All right, everybody, welcome to the Trial Site News podcast interview series. I'm Dr. Erin, your host, and today we are chatting with two scientists and researchers in Israel. We have Hadar Medini, she is a PhD candidate, and we have Dr. Dan Mishmar. So thank you guys for joining us on Trial Site News. And for our listeners, they did some really cool research on the mitochondria and COVID-19, and I'm simplifying that, but I read their paper yesterday, and it's in a peer-reviewed journal. So, but before we dive into that research, which is really interesting, do you guys mind offering a brief introduction about who you are and what you do?
I think that Hadar is the star of this show. So why don't we let Hadar speak?
Okay.
So we research on an organelle that is the power of the cell, the mitochondria, and we like to investigate the communication of the mitochondria with the nucleus of the cell, and this communication can change across different phenotypes or different diseases. And the mitochondria is a very important organelle for to investigate disease of the human many disease. And Dan, do you want to continue?
Yes, of course. I think that Hadar joined my lab as a computational biologist. And we focus in our lab, we focus on the function of the mitochondria, the evolution of the mitochondria. So in short, when you look at the next time that you look at yourself in the mirror, you will see more or less 30 trillion cells, in which in every one of them, there's a mean number of 1,000 mitochondria, and they have to communicate. These mitochondria has to communicate with the rest of the cell. And this communication is highly important for us to talk now, for us to walk, to think, to hear. It is the basics for our energy source in every movement, in every function that we do. And we were more interested in this communication in terms of regulation, the co-regulation of the genes that are encoded by the maternally inherited mitochondrial genome and the nuclear genome, which is inherited from both parents. And we asked ourselves whether such communication and so-called co-regulation is important for life. Is it if it is impaired, would it do anything to our lives? So this is one of the questions that guides our research in our lab, and this is the time where Hadar joined our lab.
Perfect. Very interesting. And did I read correctly? Are you a geneticist? Are
you asking me or Hadar?
Well, whoever wants to answer, really.
Well, I'm a geneticist. I can now formally address myself as a geneticist, but I'm also an evolutionist. I'm more interested in both the interaction between evolution and genetics.
Interesting. Okay. And what are you getting your PhD in?
Well, Hadar? Okay.
Okay, Dan. I think Dan can answer this question for himself.
For myself, I did my PhD. Now it is your turn.
Okay, so my PhD is about the single cells heterogeneity, about the heterogeneity of the mitochondrial transcript of the genetic material of the mitochondria in single cells.
Okay, very interesting. So you guys do a lot with the mitochondria.
Well, the mitochondria is our center.
It's whenever I see it in drawings, I see it as like the the smiling cell part with like the big muscular arms, like the powerhouse. So let's jump into your research. Now you looked at, and I'm going to simplify this, so jump in and correct me if I'm wrong, but you looked at the mitochondria and different cells in the lungs of COVID-19 patients, in the blood, and I believe also in controls or people or samples from people who didn't have COVID-19. Can you explain why you did that? What were you looking for? What were you thinking, like how the mitochondria relate to COVID-19 specifically?
So let me answer that, and then Hadar will dive into
Sure.
for the details. Is it okay, Hadar?
Yeah, of course.
So she's nice to me. This is the good thing about Hadar. She's extremely, extremely talented, but very nice to me, despite the fact that I'm old.
A good combination. Okay.
Yeah. So the question started, in fact, sometimes in science, you have to accept serendipity. And I hate this word, but I can tell you that we were more interested in the heterogeneity of mitochondria within cells, even cells that look the same. And this is where Hadar joined to my lab when she started working on other types of cells in the pancreas, the cells that are important for secretion of insulin, the beta cells. And she, to make a long story short, she looked at beta cells and she found that they do not look the same in terms of the mitochondrial regulation. But this did not connect to any disease. And when it comes to for us as evolutionists, it is wonderful. But if you are interested in talking to physicians about that, they are the first thing that they ask, okay, so what happens if this is impaired, if the regulation of the mitochondria and the nucleus is impaired in beta cells, does it do anything? And we could wave our hands a lot, but it didn't really relate to a specific disease right now. So we had to break to start thinking in a different way, in a similar or different way, and then look for a disease, and then ask whether this disease impairs the mitochondrial function. So we ran into a paper that was written on September 2020. A very important paper for our sake. This paper was written by a Brazilian group, and these are virologists. They took cells and infected them by various kinds of viruses, including our favorite virus, the SARS-CoV-2. We all like it, the SARS-CoV-2. We can relate to it. So in any case, they asked what would be the best environment for this virus to replicate. And they had some idea, but what they found is, to us, was astonishing. What they found is that the virus, specifically this virus, likes to replicate in glycolytic environment. This means that from some reason, the cell does not use the mitochondria as the source for energy, but the glycolysis, which is an ancient biochemical pathway to generate energy, far less efficient, but easier to accomplish. So they found that the virus SARS-CoV-2 likes to replicate, prefers, in fact, even needs a glycolytic environment to replicate. And they came with this hypothesis that the virus induces the activity of glycolytic enzymes in order to replicate.
And just to put it in easy terms, glycolysis is just the breakdown of sugar, right? And we're getting...
Exactly.
Okay, all right.
The breakdown of sugar to other particles in the end, but you get ATP, you get energy. Only 2 molecules of ATP, but the only thing that you have to do is to break down sugar, and that's it.
Right. And that's what the virus likes. It wants to
It
have
likes it very much. In fact, it needs it to replicate.
Okay.
So this was one very crucial evidence for us to say, maybe they are missing here. These people are missing one thing. Maybe the virus interferes with mitochondrial function and lets the cell choose glycolysis, and then it paves the path for the virus to replicate. So this is, you see my hands, they are moving a lot. It means that it's just hypothesis. The other thing that attracted our attention is that Hadar looked at the genome of the virus. This is a very compact genome. It encodes only for 29 proteins, but 3 out of those proteins were shown to be translocated to the mitochondria. Another thing that connected the virus to the mitochondria. And the third thing is the thing that people are talking all the time when they talk about COVID-19. They talk about the storm of cytokines, the cytokine storm. It's a word, but what does it mean? It means that the immune system sends so many signals that they cannot tolerate any more signals. Signals have to be sent in minute amounts to be understood, but just imagine yourself standing in a room full of people and they are all shouting at the same time. How can you understand yourself thinking? You cannot do that. And this is what happens to the cell. We thought to ourselves that, come on, signaling is what the mitochondria does. So this was the motivation that Hadar and I got and were urged by in order to go to do this research. And since Hadar and I did not want to isolate any virus anymore, all we wanted is to look at the data that other peoples did. Simply isolated RNA, RNAs, the transcripts that come from our genes across all the genome,
right?
Coming from, those upper respiratory tract samples, the thing that people, I don't know how to say it in English, but the thing that you shove into your nose and take samples,
right?
Just to, yeah, exactly, to see whether you are carrier or not carrier to the virus. And people also took, the same people were also subjected to be taken for the blood, to take their blood.
So just to be clear, the RNA relates to the copies of the transcript coming from the cell, our cells. It wasn't that you're not talking about the viral. Okay, we're not talking about the viral RNA, just to, right.
Exactly.
Okay. So you took these samples. What did you find?
So first, then I will let Hadar speak because she's the one who did the job.
Okay.
So the point is that people did what they did. They took samples, as I said, from these epithelial cells and from the blood. And the disease is in the respiratory tract. It's not in the blood. People go to the hospital not because of their blood, they're going because of their lungs. So we thought that if the mitochondrial function will be wrong, it will be probably in the lungs and not in the blood. So the blood was simply a control for us. How wrong were we?
Interesting. Yeah.
And the second thing is that, people do not think of the mitochondria. For some reason, they ignore the mitochondria, which is good for us. Other people do the job, and all we need is to take the garbage bins, dive into the garbage, I'm sorry about this thing, but and take the data coming from the mitochondria that just hides under the radar. And this is what Hadar did. Go on, Hadar, tell them the story.
So as Dan said, we took the samples from COVID-19 patients and from control individuals. And the samples were divided into respiratory tract, where you do like the PCR swabs for the respiratory tract samples, and the blood when you take blood from people. And then we could extract the data from these data sets and analyze it. And we found that only in blood and not in respiratory tract, we found a decrease in all mitochondrial DNA genes, a decrease in the RNA of the mitochondrial DNA genes. And this was very surprising because in the respiratory tract, it wasn't the case. So we thought that maybe some cells in the blood could affect such a phenomenon. And so we went from the bulk samples, we went into single cells. And then we thought that some immune system cell types, actually most of them, showed this decrease. And the epithelial cells of the respiratory tract samples did not show it. We saw also that the communication between the nuclear genes that are working with the mitochondria and the mitochondrial genes is impaired in the COVID-19 patients. So we saw that this was also associated with pathways, with a group of genes that are related to what we call the cytokine storm that could affect the cytokine storm in these cells.
So so it sort of went against what you thought would happen. It's like, what? You get this result. So you see this happening in the blood and not in the lungs, the respiratory tract. So could you say, well, is this a respiratory disease or is this, a disease of the mitochondria? And what does that mean?
Would you like to answer, though?
Whoever wants to jump in.
I will let you answer then.
You would like me to answer? Okay. So this is still a respiratory tract disease. After all, people are going to the when the disease goes worse, people are hospitalized because of their problem to respire. But what we think is that we underestimate the role of the immune system in this disease, because when Hadar employed her expertise in analyzing single cells RNA analysis, she also analyzed cells coming from the lung. So when she looked for cells in the lung, she saw that when looking at the lung as a whole, Hadar mentioned that before, nothing was apparent. But when she did single cells analysis, she found hiding the immune system cells, which occurs also in the lung. And they acted exactly as the cells in the blood. In other words, what she found is that the mitochondrial function, the communication between the mitochondria and the nucleus, as Hadar said, was impaired specifically in the immune system cells, regardless if they come from the blood or from the lung. And this associated with increased levels of genes that are important for, as I mentioned before, glycolysis. So practically, we are not, I don't think that we are talking here about a new kind of an immune disease. I think that the immune system plays a crucial role in this disease in terms of this cytokine storm, in and the severity of the disease, which allows for the virus simply to replicate and cause, as a terrible side effect, the lung disease. But this is a speculation, eh?
Right. So it's a theory. You need to test it. Is there anything you want to add to that, Hadar?
We also saw that in the timeline where people were infected by the disease and then they were like healthy again, we saw that in the healthier state, when they passed the disease, we saw that it was reversible. The mitochondrial RNA was like, in the convalescence people, it was like in the healthy people. So,
in other words, in people that recovered from the disease, the mitochondria came back to normal.
Okay, so that's good news.
Of course it is.
Right.
I agree.
So getting back to the cytokine storm, and you're right, those are words that we've heard a lot in the news, and bad things happen when you get the cytokine storm. If that starts with the signaling in the mitochondria somehow leads to this cytokine storm, which I'm sure you can elaborate on way better than I could ever do. What does that mean for possible interventions, treatment? Could you possibly do something that could make the mitochondria not do that, not, do the things that the virus makes it do?
So Hadar, if you are okay, I will answer that.
of course.
So I think that this is a question that I've been asked a lot. So what do we do now? So the first thing that I have to say, the best prevention to be infected by a disease is to be immunized. Is to take these immunizations, Pfizer, Moderna, whatever, who does this work. They're doing excellent work to prevent the virus from infecting our cells, and in this way they prevent, or it assists our body to simply avoid the virus. But once the virus avoids the immunization, and this is what happens with the Omicron, which is a different kind of story. I think that it is not part of this podcast. But in any case, what happens when with people that, after all, get the disease, we would like them not to be hospitalized, and we would like to reduce this storm in order to make this disease manageable by people. And in fact, one could think of how can we assist the mitochondria to get back on track, even in disease conditions. So there are many ways, but in fact, I can think of a way that, it was not very much tested, but people like antioxidants. Antioxidants are the ones that we drink when we drink orange juice, vitamin C, vitamin K, CoQ10, all kinds of things that could be taken from, without any prescription from the shelf.
So antioxidants.
Yeah.
I mean, so you get those from your diet too.
Exactly. So
just the antioxidants that will help. Diets or supplements.
Oh, this would be an exaggeration. I think that it has the potential, but after all, if you take something to your mouth and it goes through your system, and then it goes to any kind of cell of the body. So I think that if people are going to start thinking about antioxidants as not just mere dietary supplements, but a way to design these antioxidants to get to the disease cells preferentially, they might have the potential to reduce the storm. and in this way, to reduce the severity of the disease. But, it's a
Can
suggestion.
you explain how, and I know that's there's complicated, there's reactive oxygenated species, but if I'm taking an antioxidant and I say, oh, it's going to help my mitochondria function better, what are like how will it do that specifically? If there's an easy way to
No, there is an easy way.
Okay,
In fact,
okay.
The mitochondria, you could imagine the mitochondria as an engine. After all, this engine works in a way that, you transfer electrons, you pump protons. So you pump protons, transfer electrons, and generates energy. That's it. It's like a football match. You generate energy by just pumping things, transferring electrons, making energy in the end. But, like an engine, this engine works and generates so-called smoke. And the smoke is the reactive oxygen species. But this is not just mere smoke. When your car starts smoking, you want to get it to the garage. But the point is that the mitochondria generates in purpose this smoke. And these reactive oxygen species are not just byproducts of the energy production. These are signaling molecules. Now, if they are generated in the correct amount, they transfer signals. But if they are generated, you get so many signals that the cells does not know what to do first. So the bottom line is that you would like to reduce the amount of these signals and to a degree that the cell can comprehend what it's working with. That's the point of working with antioxidants. It's not just putting anti-aging cream on your body and hope for the best.
Hope for the best.
or make these people earn more money, but this is another issue.
No, that was a great explanation.
Yeah.
I think that a lot of people will be able to grasp that. And when you guys mentioned at the beginning of the podcast, the mitochondria's role in disease, it's not something that you're right, we don't hear a lot about this. So it makes you wonder, what are the role, that the mitochondria is playing in other diseases, viral or not? Do you guys think it's more obviously more significant than, I guess?
Well, the mitochondria sits in such an important crossroad in the cell, in every cell of our body. And when you come to think of that, there are many diseases, mainly neurodegenerative diseases, such as Alzheimer's disease, Parkinson's disease, ALS, and others, in which you find mitochondrial dysfunction. In fact, the hallmark of Parkinson's disease is a specific mitochondrial dysfunction in one of the protein complexes that works in the mitochondria. And this is not only neurodegenerative diseases that have something to do with the mitochondria. Diabetes has a lot to do with the mitochondria, type 2 diabetes. And many types of cancers were shown to associate with mitochondrial dysfunction. And this is only a partial list. So when you come, there's a problem here. If you find a thing that is involved in everything, then you start thinking, oh, come on, this is impossible. It's not happening to me. I don't want to work with a thing that is such as complicated. Let's choose another topic. This is one way. The other way is to say, yes, well, it is central. So when you deal with the mitochondria, when you interfere with such a very fundamental mechanism for our lives in every cell of our body, then all our body will be torn apart. Well, this is too much, but will not function very well.
Absolutely.
So, yeah. And this is why the mitochondria is not only important for disease. I have to say that. It's not important only for disease. It's also important for the emergence of new species on this planet.
Oh, wow.
Oh, yeah.
That's an interesting
complex.
tangent. I kind of want to go down that road a little bit.
Would
We
you
think
like
it
to?
All right, let's take a couple minutes and explore that. Just
Yeah.
now that you're here. So
Would you
Yeah.
like to hear
Yes.
about this?
yes. If you have the time.
Hadar, I'm taking too much of your time, Hadar.
No, it's okay. We talk, we listen.
This is true. So this is what we do. We talk and listen. This is what. So in any case, the mitochondria is with us for the past 2 billion years. This is a long time. In fact, I cannot comprehend such numbers. It's an extremely long time. And when you come to think of that, the mitochondria was once a free-living bacteria. And it was fused with an ancient creature, which, it has a name, an archaea. It was fused to create what we know as cells with a nucleus. We call it eukaryotes. These are big names, but only eukaryotes are the organisms that gave rise to complex organisms. Bacteria always remains unicellular, archaea only unicellular, but eukaryotes could be unicellular and complex like us. Well, you are more complex than me, but this is another question. Us, the Drosophila, I mean the fruit fly, the cabbage, every organism that has more than one type of cells is a eukaryote. And these eukaryotes rely on the existence of the mitochondria and the interaction between the mitochondria and the nucleus, because this fusion that created all eukaryotes was associated by loss of independence. We cannot grow mitochondria outside of the cell, and we cannot grow the cells without the mitochondria. So just imagine, if you think about the mitochondria and us as the longest lasting marriage ever, then how can you think of these two entities as separate entities? We're not separate. We have to communicate. We have to co-regulate. And this connection has to be such a long connection should last for such a long time, because it co-evolves. When a change occurs in one, it will be compensated by the other. But if it does not occur, it breaks. If it breaks, one way is to get a disease, but the other way is to create new species on Earth. A long explanation for one thing. Yeah.
That was a great explanation. I love that. I love that stuff. So hopefully some of our viewers like it too.
The two viewers that remained after all these things. Huh?
Thanks a lot, Aaron, for taking. Go ahead.
Also, the mitochondria is inherited only by the mother to all of her children.
Right.
So it's a nice point.
Yeah. No, absolutely. It's very interesting. I have two more questions. And these will be brief, I promise, because I know you guys have stuff to do.
Go ahead.
And I have stuff to do too, but it's interesting. I like this stuff. In addition to antioxidants, are there any other ways that somebody could improve mitochondrial function?
Well, antioxidants are it's working with the outcome of the mitochondria.
Mm-hmm.
The other way is to improve the connection and the communication between the mitochondria and the nucleus.
Mm.
And in fact, this is a thing that is not very well understood, because one way to connect two organelles in the cell, the mitochondria and the nucleus, is to send signals. And I mentioned the reactive oxygen species as one type of signals. But the other way is to send proteins that are regulating the function of other proteins, and to send them both to the nucleus and to the mitochondria. And this is a matter of debate because the dogma that exists for the past 50 years states that the mitochondria is regulated separately from the nucleus, and the only way to connect to communicate is by sending signals. Suppose that we are that we have to argue with this dogma, and there are some proteins that exist both in the nucleus and in the mitochondria and regulate both. If we target these, we will assist the function of both organelles and assist the cell. Does this answer the question?
Yeah, no, it neat stuff. I'm learning a lot. So thanks
Excellent.
to you both for coming on. I guess my last question is, how are you guys feeling about COVID-19 right now in your country? Is it things getting better? Are you hopeful? You're
Hadar, please start.
I think we should all do vaccines. No, we should be careful and stay healthy and do whatever we can to stay healthy and be safe, and for our family, for our friends, and for our community. And yeah, we hope it will get better in the future.
Yeah, yeah. Seems to be the going around in circles a bit, but yeah, well, we have to, I think. And I guess eat your antioxidants. That can't hurt, right?
It can't hurt you at all. In fact, it might improve your situation in unexpected ways.
Mm-hmm.
But I think that Hadar was very much right. We cannot stop our lives because of this virus. We have to protect ourselves. And she mentioned it very nicely, Hadar. One has to immunize yourself. You have to protect yourself, but continue doing research. This is what we are doing. This is what we need to do in order to maintain not only our mental health, but also to provide some hope for others.
Yeah.
This is my thought.
No, I agree. I've picked up some new hobbies. Well, things aren't. I play my violin. I enjoy it. My neighbor, not so much, but there we are. So.
No, you're fantastic. I have to hear that. I like music very much.
Yeah, no, it's just living with neighbors and everybody's home. Anyhow, you guys, thank you so much for your time today. Very interesting research. I look forward to sharing with our viewers and hearing their comments. If you have any more research in the future, please let us know at Trial Site News so we can share it with everybody. Great to connect, and best of luck to you both and everybody in getting out of this pandemic mess.
Thank you so much.
Thank you.
It's been a pleasure.
Absolutely. Okay, guys, enjoy the rest of your day.